Our second keynote speaker will be Amy Howard. Amy Howard is a postdoctoral researcher at the Wellcome Centre for Integrative Neuroimaging at the University of Oxford. She also did her PhD there. Her research focuses on combining MRI and microscopy data to both validate and drive biophysical modeling of the brain tissue microstructure. Dr. Howard, please proceed with your talk titled Mapping MRI to Microscopy.
Fantastic. Just to check you can see my screen properly.
Yes, that's great.
Yeah, awesome. Thank you so much for that introduction and also for the opportunity to speak here today. So I'm going to be speaking on kind of more generally mapping MRI to microscopy, and I'm actually going to change the title slightly to add this byline from monkey to man, as I thought I'd take the opportunity to talk about two projects that I think are really exciting coming out the lab. So we'll start off with some macaque data and then move on into some human tissue. So with that, let's get started. So as we've seen throughout this conference, having MRI and microscopy in the same tissue can provide unique opportunities for multi-scale neuroscience, mapping microscale cellular structures to macroscale MRI signals that can ultimately be acquired in vivo. I'm just going to move you all. 1 second, there we go. Fantastic. So to this end, we've recently created a new open access resource called the BigMac dataset. BigMac includes in vivo MRI, postmortem MRI, and multi-contrast microscopy in a single whole macaque brain. The dataset is fairly ambitious in scale, so we have in vivo data acquired over multiple scan sessions, around 270 hours postmortem scanning, and well over 1,000 hours of microscopy, resulting in around 4 terabytes of raw data. So let's unpick this a bit. So the in vivo MRI includes structural, task, resting state, and diffusion MRI, whilst postmortem we have structural, T1 maps, and an extensive diffusion MRI protocol I'll come on to in a couple of slides. And this really provides us a fantastic opportunity to link our postmortem MRI and microscopy to in vivo data in the same brain. And this is really trying to overcome one of the key challenges, I think, with these kind of validation studies, is that our quantitative MRI parameters are often considerably different postmortem and in vivo. So we always have this challenge of how do we translate our results into in vivo and eventually human cases. The postmortem diffusion protocol includes data at 2 spatial resolutions, so 0.6 millimeter with a b-value of 4,000, whilst at 1 millimeter with b-values of 4, 7, and 10,000, where in the outer 2 shells we have 1,000 gradient directions, making this, to our knowledge, the highest angular resolution dataset yet acquired. The high spatial resolution allows us to characterize fiber populations with fewer partial volume effects, whilst the 1 millimeter data allows us to retain good SNR when we push to the higher B values, and also we can characterize these or acquire these very large number of gradient directions in a feasible scan time of just over a week. Further, we have both linear and spherical tensor encoding to support more advanced microstructural models. And of course, with a high B value, we obtain beautiful diffusion contrast. So here I'm just showing different raw diffusion weighted images as we trace out this spiral on the Q space sphere. All our data was acquired on a 7 Tesla small animal scanner using a spin echo multi-slice sequence with single line readout. This sequence is beneficial because we have very few image artifacts, but it is fairly slow, resulting in around 270 hours scanning time. So in BigMac, we want to relate our MRI to microscopy data, which has both high spatial resolution and high specificity. So what microscopy data was acquired in BigMac? After scanning, the brain was sectioned along the anterior-posterior axis, where consecutive slides were acquired for one of 6 different microscopy contrasts, so either polarized light imaging or histological staining. Each stain is repeated every 350 microns, and we have whole brain coverage. So far, we've acquired 3 sets of data, where here I'm just showing example images, and we have whole brain coverage. So on the left, we have polarized light images, where we utilize the inherent birefringence of myelin to estimate the primary fiber orientation per pixel, where here we show the orientations in 2D using this 2D color wheel, and the data was acquired with a resolution of 4 microns. We can see beautiful orientational information within the white matter and also extending into the gray matter, as well as through subcortical structures, which can be difficult to characterize using diffusion MRI tractography. In the middle, we have a Gallius silver stain where myelin has been stained brown, where we can again visualize myelinated fibers, but also characterize myelin variation across the brain. And on the right, we have a crescent violet stain where the cell bodies of both neurons and glia have been stained purple. And we see this beautiful variation across cortical layers. The histology was acquired with an order of magnitude higher spatial resolution of 0.28 microns, meaning that we can visualize single fibers and even individual cell bodies. So the polarized light imaging data is inherently already quite quantitative, so we can directly compare that to the MRI. But the histology, we need to try a bit harder. And this is because in histological data, we see variations in the background staining, meaning that our staining densities are only semi-quantitative. And what I mean by that is that here you can see there is a region where we have very faint staining, even though there are plenty of cell bodies. So how do we make sure that we're sensitive to the cell bodies irrespective of this kind of background staining? So in our lab, we've developed methods for data-driven stain segmentation, where we extract small patches of the histology, and in a data-driven manner, we develop thresholds that will segment the positive myelin from the background tissue. We can then calculate the number of positive pixels over a local neighborhood size of around 150 pixels to estimate the stained error fraction. We can repeat this across the entire slide to produce these maps of stained error fraction that faithfully represent myelin variation across the white-gray matter and across the subcortical structures as desired. We can perform similar segmentation for the Nissl stain slides, where here we can even go further and try to segment single cells, at least to the ability of our eyes, which allow to extract morphological metrics, such as approximate cell area, circularity, the number of cells, the radius, or the nearest neighbor distance, all of which may relate to our MRI in interesting ways. A second type of quantitative analysis we can do is structured tensor analysis, and this allows us to estimate the primary fiber orientation per pixel, again on the 2D plane as shown by the color wheel. We can of course do this for our myelin stained slides where we can visualize myelinated axons, but interestingly we can also do this for the Nissl stain as the cell bodies tend to align along the fiber orientation. Where both types of data are even able to visualize these interesting details such as these interdigitating fibers entering the corpus callosum. So if we want to then relate this microscopy to our MRI, we need to co-register them. And by that, I mean we need to warp our 2D microscopy slide into our 3D MRI volume. And this is really hard. We're trying to link data that is 3 orders of magnitude in scale different from microns to millimeters. Our images often have really notably different contrast. There are a ridiculous number of degrees of freedom in this optimization. And also the microscopy can have artifacts from tissue processing, such as shrinkage, tears, bubbles, etc. In our lab, we luckily have the wonderful Istvan Hazar, who's developed a tool called TIL, Tensor Image Registration Library, that was specifically developed to kind of overcome these challenges of MRI microscopy co-registration. In BigMac, we use a 2-stage registration process, where in stage 1, we register each microscopy slide individually to the MRI, and in stage 2, we optimize over all of our slides simultaneously, as we found this was could increase the precision of our registration. Utilizing these high-quality co-registrations, we can now take our microscopy metrics and start to build up these 3D microscopy volumes, where here I'm showing an example of the Nissl stained error fraction. Even though each slide here within the coronal plane represents data from a different microscopy section, we can see that our metrics vary smoothly across the brain, suggesting that our slides can now be combined with minimal impact from these artifactual staining variations. Further, our volumes describe the microstructural variations of interest across the white-gray matter, across cortical layers, and across subcortical regions as we would desire. What's interesting here that I'd just like to highlight is this increased Nissl density in the posterior brain, which relates to one of the previous talks where they found similar results in big brain in humans. But here, of course, we don't have a single microscopy volume. We can have many microscopy volumes relating to fiber orientations, myelin and cell body density, as well as these morphological metrics, such as the number of cell bodies or cell body circularity. Okay, so we now have our microscopy and our MRI within the same space, and we have whole brain coverage. What are we going to do? Well, we need to define different axes along which we want to do meaningful comparisons. So, of course, we can do voxel-wise MRI microscopy correlations, where here I'm showing correlations between different microscopy metrics and a range of diffusion metrics that are extracted from our rich diffusion protocol. And in the middle, you can just see the correlation coefficient. So we can instantly see that the MRI has variable sensitivity to these different microstructural features, and we can start to pick out specific relationships, such as the DTIMD seems to have a strong correlation with the nearest neighbor distance of cell bodies within the white matter. Of course, there's a huge amount of information in here we can then go in and probe. But we can also ask if there are other interesting axes. So for example, I can run tractography to define different white matter tracts. I can then plot the Nissl stain diffraction, which we can interpret as the glial density within the white matter across these different white matter tracts, where we see interesting variation across the brain. We can then ask, does MRI show similar patterns? Well, if we correlate with structural MRI, we see a fairly good relationship. If we correlate with different diffusion metrics, we can see that the kurtosis, the third parameter, also shows a slightly better relationship. Now, interestingly, and this is a really recent result, so I need to go back in and check this, take it with a pinch of salt, but the best correlation I found was this isotropic diffusivity from combined linear and spherical tensor encoding, which has this very, very strong correlation with glial density across the white matter. And so, of course, I need to verify, maybe double check this isn't being driven by things like partial volume effects, although my initial investigations suggest that it is not, which mean that perhaps we could conclude that we could use these more advanced metrics to, for example, investigate glial changes across the white matter. We can, of course, define other axes of interest, such as cortical profiles, where here we can plot microscopy or MRI metrics from the white grey matter boundary up to the peel surface, and we can compare them along their sensitivity to different cortical layers. Or we can, of course, project things onto the surface and again, project them there, where both of these types of analyses are kind of ongoing work within the lab. Finally, I just wanted to mention that, of course, we don't need to do a direct comparison. We can also use this kind of whole brain microscopy data to quantify variations in cellular distributions across the brain or across regions of interest, where here, for example, I'm showing the approximate cell body radius across the white matter, temporal cortex, and motor cortex, where we can see the data faithfully represents the small glial cells in the white matter, as well as this a small number of large BET cells in the motor cortex. And we can then use these kind of variations in cellular distributions to inform on more microstructurally realistic simulations of MRI to test MRI sensitivity to specific tissue features. Okay, so that's kind of all I wanted to say on BigMac today. I hope that I've convinced you that BigMac includes rich MRI spanning tissue states from in vivo to postmortem, and MR contrasts from functional to structural to diffusion MRI. Furthermore, we have quantitative microscopy metrics to characterize the cyto and myeloarchitecture across the brain. We believe that there's a whole host of applications for this kind of whole brain multi-contrast data, from neuroanatomical investigations to multi-scale neuroscience, to MRI methodology and validation, perhaps of interest to people here, as well as machine learning and data fusion. Our data is made openly available via our online platform called the Digital Brain Bank, and BigMac also comes with a fairly extensive online documentation, and example code and tutorials to get you started. So please do get in touch if you think this data could be of interest to you. But since we're on a conference that's got a focus on neurodegeneration, I thought I would kind of change gear for the second part of the talk and talk a bit about a different data set that's also available on the Digital Brain Bank, but here under the theme of digital pathologist. So this data set, I'm going to preface by saying it was largely, the work has been led by other members of the lab. So Manuka led a lot of the data acquisition, Istvan did the registration, and Daniel did the microscopy curation and a lot of their MRI microscopy correlations that I'm going to show today. So I'm really presenting on their behalf. So this is a pretty amazing data set where we have donated human brains that have been kindly donated to the Oxford Brain Bank. And we have brains from both patients that were diagnosed with motor neuron disease and also healthy controls. We first acquire whole brain postmortem MRI, including structural, relaxometry measures, DTI, and QSM. We then take the brains, we section them, and we acquire small tissue blocks from different regions, such as the motor cortex, the visual cortex, frontal regions, and white matter regions, such as the corpus callosum. And the reason why we sample these different brain areas is that MND has a very well-described spread in disease pathology. So it starts off by affecting the motor cortex and then will kind of spread anteriorly, posteriorly across the brain, meaning that within a single subject, we may have some brain regions that have quite severe pathology, whilst other regions can basically act as a healthy control. For each tissue block, we then section them and stain them with different stains that are sensitive to both myelin. This is ALS pathology, neurofilaments, microglia, and activated microglia, which is a biomarker of neuroinflammation. And I think this is really one of the strengths of this data set, is that we have these multiple stains within each voxel, allowing us to build up this more complete picture of the tissue microstructure. We can see whether across voxels there are changes in myelin, neurofilaments, microglia, and that allows us to start to pull apart this difference between MRI sensitivity and specificity to different tissue features. And what I mean by that is not only can we detect that there has been a change in the tissue, but we can specifically relate it to either demyelination or axonal loss, where demyelination is going to affect our myelin stain, and axonal loss would affect both myelin and neurofilaments. Okay, so in this data set, we first take our tissue blocks, we stain them, we then develop automated methods to extract the stain error fraction again. We register them to the MRI using TIL, and that allows us to then perform these MRI histology correlations, where each data point is a different voxel, and we've combined across brain regions and across subjects, so that we have many voxels driving these relationships. With this data, we can perform a simple correlation analysis, which is just like what we normally see, where we see lots of very well-described relationships, as well as some that are perhaps a bit more surprising and actually echo really nicely with Sylvia's talk previously, where we see this DTI sensitivity to activated microglia, which is this biomarker of neuroinflammation. But because we have multiple stains, we can start to move on to try and do something a bit different. So why don't we try and do partial correlation analysis, which can try and get at this question of MRI specificity. And partial correlation analysis, for anyone who needs a reminder, is where we analyze multiple variables simultaneously, which allows us to account for their covariance. So we can basically treat one variable as a confound. So let's take a toy example. We have an explanatory variable, such as large feet, and an observation of people who find aeroplanes uncomfortable. If we did simple correlation analysis, we may find that these two things positively correlate, from which we could maybe, not with our heads on properly, conclude that big feet maybe means you're generally less comfortable on airplanes, which is true. But is this really related to feet? Well, if we run partial correlation analysis and we account for the covariance with tall people, we'll see that this correlation coefficient will basically drop to 0. Whereas the correlation coefficient for tall people, whilst accounting for large feet, will remain, meaning that we can then conclude that comfort is more specifically related to height, and the correlation that we saw with large feet was due to this covariance of people's height with their foot size. Tall people generally tend to have large feet, right? Okay, so let's go back to talking about stains. Here we're going to analyze multiple stains simultaneously. So we'll correlate one stain whilst accounting for the co-localization of another. The results I'm going to present are fairly complex, so let's build it up line by line. So on the top here, I'm showing the correlation between these different stains, histological stains, an FA on the left and MD on the right. And the top line is just the simple correlation coefficient that we saw previously. Let's see how the correlation coefficient changes now as we account for both myelin and neurofilaments. So if we zoom in on this green box here, we can see that originally we have a strong correlation between myelin and FA, which when we account for neurofilaments doesn't change so much. When we, to start with, we have a strong correlation between FA and neurofilaments, which then when we co-localize for myelin, we can see it basically vanishes to 0. And from this, we can conclude that at least this data suggests that FA is sensitive to myelinated axons rather than neurite load in general. And that if we think about the difference between the myelin and the neurofilament stain, we can then think that changes in the unmyelinated axons and dendritic load does not contribute to FA. And here, of course, we're not accounting for dispersion, we're just looking at the general load of neurites or myelin. Interestingly, when we look at MD, we see a similar effect where the correlation coefficient reduces, though it's not 0. We again see a similar effect on R2 star. And on R1, we actually see the opposite effect, suggesting that R1 is sensitive to neurites in general rather than myelin alone. And I wanted to highlight this kind of analysis because I think it demonstrates the pitfalls in some simple MRI histology correlations. In simple correlation analysis, we may erroneously conclude that, for example, FA is affected by neurites in general, whilst our partial results show that FA is primarily related to myelin with little or no sensitivity to unmyelinated axons or dendritic load. And it's really the spatial covariance of the microstructural features that may lead to these kind of misleading relationships. Okay, so let's continue. We'll now co-localize for microglia and also activated microglia, where here, if we can see that there's again a similar relationship where the correlation with just the purely microglial stain basically vanishes to zero, suggesting at least here that DTI is sensitive to activated microglia or neuroinflammation specifically, but not just the microglial load in general. We see a similar effect for R2 star and no real effect for R1. Okay, so then we can co-localize or partial out the effect of all stains simultaneously. And I really want to just highlight this result here, where we can see that even accounting for changes in myelin, so demyelination, neurofilament, so axonal loss, or just the general presence of there being microglia in the tissue, we still see this DTI sensitivity to activated microglia. And what impressed me here was that if we then change to white matter only analysis, this correlation, fairly strong correlation, remains. So it suggests that it's not, this effect is not being driven by kind of gross differences between the white and grey matter tissue composition. Cool. So this suggests that I think this result's really nice as well, because it kind of talks to the previous talk, that DTI is highly sensitive to neuroinflammation after accounting for all other microstructural changes, such as demyelination, axonal loss, or general microglial infiltration. Excellent. So in the interest of time, I'm going to wrap up there. Just to conclude this section, today I presented quantitative MRI and multi-contrast histology from donated human brains across multiple subjects, where this data is all made openly available. So please, again, get in touch if you'd like a copy. The fact that we have multiple stains allows us to run both simple and partial correlation analysis to probe MRI sensitivity and specificity to different aspects of the tissue microstructure. Our results emphasize the need to simultaneously analyze multiple stains when validating MRI to avoid misleading inference due to the spatial covariance of microstructural features. And we find DTI sensitivity to activated microglia a biomarker of neuroinflammation, irrespective of other microstructural changes. If you'd like more information as well, please do check out Daniel's lovely publication last year in Neuroimage. So with that, I will conclude my kind of tour de force of mapping MRI to microscopy from monkey to man. And I would like to thank all of those who have been in this like highly interdisciplinary project, particularly Daniel Kaur, whose work I presented today, Istvan Hazar, and also Manuka. And with that, I'd like to thank you for listening and welcome any questions.
Thank you very much. Beautiful pictures, super cool stuff. Any questions, Sajad?
That was great, thank you. We have a few questions. The first is from Diane Patterson. Is the data available through Data Lab? If not, do you have plans to make it available through Data Lab?
No, so particularly given that we're either talking about animal data, non-human primate data, or also human data, due to kind of ethical and funders requirements, all our data, you do need to sign a material transfer agreement. It's very straightforward, but it means that we have to remain the hosts of that data before people make sure that agreement, and it's just there to ensure the data is used for kind of scientific means and that it's used in accordance to the kind of funders requirements. So we hope that we're really trying to minimize any barriers to engagement, but the way to get it is to just get in touch with us and we'll guide you through that process. It should be pretty streamlined.
Thank you. Next question. Do you have a functional MRI data from the same animal? And if yes, which kind of tasks have you used?
Yes. So BigMac, we've got functional MRI over multiple scan sessions. I will put my hands up and say that I wasn't involved with this data acquisition. So it was acquired, like I stepped in the kind of post-mortem stages. But it's it's from our collaborators who are very interested in decision making. So they're kind of, my understanding is their decision making tasks, I think, to do with like different dots on the screen, and the monkeys will get rewards, which is generally fruit juice. and they do a little tap depending on the making a decision between kind of two choices. So I believe that's what the data is. That is also made available, particularly the Z-stat maps. They're very easy to share. So we can have kind of pre-analyzed data if that was of interest as well.
Great, thank you. Another question is from Jean Chen. Great talk. Can you mention sources of signal differences between MRI and microscopy, like absence of circulating blood in postmortem? And talk about how big of a difference this can cause in your results.
Between MRI and between in vivo and postmortem MRI, or between?
Between MRI and microscopy. But I think it's like in vivo and ex vivo probably.
Yeah, so the tissue changes considerably post-mortem, right? So we have like the death, and there will be cellular changes. One of the benefits of working with non-human data is that you can have perfusion fixation, which means that you can fix the tissue much sooner, which really can mitigate some of the kind of neuronal death that will otherwise occur very, very quickly after death. But obviously fixation still makes changes to the tissue microstructure, so that is important to take into account. I think, you know, it's really by having in vivo data that you can also compare to the microscopy that we can start to kind of pull apart these effects. So we have done some comparisons like taking kind of diffusion estimates of ODI, correlating them just from the postmortem data with the microscopy, and then running the exact same analysis with the in vivo data using the same models. And what we found was that the relationship was extremely similar, but there was a kind of shift in the kind of offset. So as we all know, like diffusivity, pure diffusivity values will change considerably post-mortem, but the kind of sensitivity to variation across the brain was very similar.
Thank you. Maybe one last question from Olivier. Could BigMac be used as a benchmark to test the biological specificity of other diffusion modeling techniques like NODI or SMI?
Yes. So I was always very nervous of running NODI post-mortem because if people are familiar with NODI, you have to set an axial diffusivity. And that's contentious enough in vivo. Post-mortem, you really don't know what your diffusivities are very well, right? So NODI, and depending on how you set that, like significantly changes the output of NODI. So I was nervous of running it post-mortem, but some of the kind of newer methods to run the standard model, you don't need to make assumptions on diffusivities, for example. So, and we have the data that can support those models. So yeah, 100%, it would be really good to, like today I kind of presented DTI and DKI analysis because often that still has most clinical relevance, right? They're things that are really being used routinely in clinic. But as things are progressing more towards a standard model, yeah, we should definitely be running it on BigMac data.
Okay, so I have one or two more questions, but I think we can move on and I can ask them the discussion. Thank you.
Thank you so much.
Okay. All right. Thank you very much to both of our keynote speakers.